Hi everyone, welcome to the session today. We're very happy to have Christophe join us here. Hi Christophe.
Hi Lan, thanks for having me. I'm Christophe Bodenreider and I'm currently serving as the Associate Director for the Target Translation Consortium at EDDC.
So in this short segment, we will discuss a key step in the drug development process, target validation. So Christophe, could you please share with us what is target?
The term target can be vague. In the context of this discussion, we consider as target any molecular entities present in human. The most covered are proteins, such as enzyme receptors or ion channel, but also signaling proteins such as cytokines.
So it means the researchers actually looking beyond just the proteins, right?
You are right, Lan. For example, some antibiotics block the function of the bacterial ribosome that is made of nucleic acid. In addition, mRNA and microRNA are more and more considered as suitable drug targets.
So if we put it in the context of the drug development, how do we link the target with the disease?
All drug discovery projects start with a scientific hypothesis. When a target X or a biological pathway Y is perturbed either negatively or positively, it is translated into an effect on a given disease.
But until then, it's only a hypothesis, right? When do you need to validate it?
So in fact, the validation of a target happens right at the beginning of the discovery process. Or I better say the preclinical validation, where we use in vitro and in vivo models. In fact, the final validation only happens in clinical trial in human.
Okay, so if it happens in the beginning of the drug development process, can I understand it in a way that it actually affects both positively and negatively of the entire process?
That's right. The fact that lack of efficacy is the major reason for drug failures in clinical trial show the challenges of target validation. Target validation lays the ground for the entire drug development. It is at the start of a process that can last for 10 years and cost up to $1 million. Therefore, you better make it right at this stage. You can change many parameters during the development of a drug, except one thing, the quality and the validity of the target.
Okay, so now we understand that the target validation is really a crucial process. But then the question is, how do you validate the target?
So the way we validate the target has changed tremendously over the years. In the past, targets were validated based on limited number of evidences. For example, if the genetic deletion of the target changed the disease phenotype, it could be enough to proceed further. Similarly, the simple observation that the target was overexpressed in the disease cell also constituted a green light to start a drug development program.
What is the situation now with the technologies at our disposal, such as the CRISPR interference RNA or even more advanced phenotypic screening method?
Yeah, with the technology you just mentioned, the researchers now have switched gears regarding target validation. And instead of one evidence, they are looking at the array of evidence. Single observations are not considered solid enough.
It seems like a never-ending process. How much proof do you need in order to consider your target as validated?
Lan, that's a key question. In fact, the best way in target validation is to try to prove yourself wrong. Basically, you run an experiment to invalidate your target. At the end of the day, your validation works and your invalidation fails, then your confidence in the target is much bigger.
So what can help target validation?
So having a small molecule inhibitor, or tool compound as we call it, or antibody against the target can greatly accelerate the validation. These agents are not only helping for the validation, but also show that the target is druggable. What we mean by druggable is that an external agent can modify the activity of this target.
So once we have accumulated the proof of importance of the validation, how confident are we in translating it into efficacy in human?
Well, that's a $1 billion question. In fact, regardless how good you've done your target validation, you pretty much depend on the quality and predictivity of the preclinical models you're using for your validation. I take two examples. I'm very familiar with the drug discovery for anti-malarial. I can tell you that for anti-malarial, the models are so predictive in vitro and in vivo that if you conduct your target validation in malaria, you can be 99% sure that it will translate in men. On the contrary, if you take the example of Alzheimer's disease, the final outcome of target validation will remain uncertain until the end. Because the underlying causes of the disease are not well known, therefore the preclinical models are not predictive.
Well, that's great learning. Thank you very much, Christophe.
Well, thanks Lan for having me, and thank you everyone for listening to this short segment.